Thank you very much for the invitation, everyone. I'm Adam Williamson, and I will be talking about applications of temporally interfering electric fields in temporal lobe epilepsy. So the majority of my work and research focuses on therapies for epilepsy, and I'll discuss this TI aspect. I think this is probably the future of brain stimulation, and I'll discuss this in detail today, specifically focused on epilepsy. So, a small introduction about me at the moment. I am at the Institute of Systems Neurosciences for Aix-Marseille University. We are currently, or were currently, on the 5th floor of the Faculty of Medicine. However, with some of my new grants, we have now expanded to the 5th floor of a second aisle. So now I have numerous labs over here and some more students. Absolutely beautiful campus. I really can't say enough about this campus. We are connected directly to the hospital where we work with our clinicians and have human experiments. Just a phenomenal facility in Marseille. That said, I now have a second appointment at the Department of Medicine at the Karolinska Institute. So, as I was saying briefly offline before we started this presentation, the weather in Stockholm is not as nice as the weather in Marseille, France. And the food, obviously, and the wine and the cheese in France is fantastic. But really, it's pretty unrivaled to have the opportunity to participate and collaborate at the Karolinska Institute. So this is really, really world-class stuff. The facilities here are phenomenal. Really a pleasure to now be expanding to the Karolinska. So I will briefly say this about my previous work. If you were to search me on the Google Scholar and look at all of my publications over the last, let's say 10 years, it just looks like brain-machine interface using polymers, right? Most of this is these conductive polymers, in particular P.PSS. So this is more, let's say more precisely, previously, my work was, let's take probes, we'll interface with the brain directly. So we're implanting devices. And the selling point of all of this previous technology, and mind you, I'm using this as an introduction to the temporal interference, very specifically as an introduction. The previous selling point of these technologies was, if we take a look at this image in the top left, we would say, okay, I implant the brain, and in a traditional inorganic system, like a metal system, an oxide system, or I have an electrode system, I'm interacting with neurons, I have fixed interfaces. You have ions that are by no means penetrating silicon dioxides or metal layers. They're not penetrating platinum layers. They're pulled into and out of, say, for example, in the form of stimulation, capacitive double layers at the interfaces. In the case of these very unique organic polymers that I'd been previously using, the selling point was that in the organic world, the sizes of these long chain polymers is such that it allows ion penetration into these polymers. And we would then utilize this volumetric effect. So there's not really a capacitive double layer. You can have significantly larger capacitances, say for reducing impedance when you're recording from electrodes or when you're stimulating from electrodes, via covering specific traditional metal electrodes with these conductive polymer systems. And there's a very funny picture down here in the bottom right. Obviously, in the inorganic world, it's significantly more difficult to make flexible substrates that would do something as ridiculous as this. Now, this is an extreme example. For a brain implant, you wouldn't be crushing your probe to such a wild degree, but it is interesting to say that when you do crush these organic probes on these flexible substrates, all of the electrodes and transistors will work perfectly. It's been demonstrated. And so we used to say ideal for neural interfaces. And in particular, in my previous presentations, I would really stress this for stimulation. So today, though, I'm going to make the argument for temporal interference. This is something I came across not so long ago, and I think that this is probably one of the most phenomenal methods of brain stimulation, particular deep brain stimulation, using topical electrodes that I've ever seen. And so I really want to make the case for this, and I have most of my research right now focused on these TI fields. So today, this is going to be the one of, let's say, I have 3, maybe 4 slides that I will show that are not mine. The rest of the slides are results specifically from my lab. And in the slides that I show that are not mine, it's simply an introduction to a topic, and then I'll expand on the topic using our own results. In this case, it's the now classic 2017 paper by a very good colleague of mine, Nir Grossman. He is currently at Imperial, where we're talking about temporal interference for brain stimulation. The general idea is that we have one electrode, let's say in the blue system here, and it's running at a frequency f. F is some frequency that is way above Usually, we're talking kilohertz. Let's say it's 2,000 kilohertz. A frequency in kilohertz that is way above the ability to cause neurons to depolarize. And there's all sorts of reasons for neural capacitance and membrane capacitance for this, but we won't get into the minutia of the membrane mechanics. Let's just simply say, practically, no one stimulates neurons at 2,000 hertz. Now we take our second electrode system in black, and we would stimulate again with 2,000 hertz, but we would add this delta F. We add some frequency offset. This frequency offset is a traditional neural stimulation frequency. And in the introductions for the audience members here, I heard some of you talking about brain-machine interfaces and stimulation applications. You for sure are stimulating with things that are not 2,000 hertz. You're stimulating with things like 5 hertz, 10 hertz, etc. So this could be 10 hertz then. So we now have 2,000 plus 2,010 hertz. Dependent on the geometric configuration of these electrodes, and I'll show to some degree the ratios of currents, you'll have points in the field deep below the cortex where you have an alignment of the electric field components. Let's say that this is the Z direction, this is the Z component aligning, where you have complete interference. You have constructive and destructive interference. And very similar to electrical engineering and communications theory, where you have carriers and envelopes, you end up with this carrier envelope system. You have, say, the black electrode providing the 2005, the blue electrode providing the 2000, and they will come into and out of phase at a rate of delta F. So we now have this delta F. And experimentally, my friend Nir Grossman showed that you can depolarize some neurons subcortically using this envelope, even though there is no 5 hertz, 10 hertz, 20 hertz. That frequency just simply isn't there in the PSD of the signal. This envelope, this beat frequency, does cause the neurons to depolarize. So how is this interesting for us specifically in epilepsy? I will be talking a lot in the first couple of slides about a publication I've been working on for a couple of years, which was very interestingly just published online a few days ago. But it's an orientation dependence of this TI. We're going to become very specific with the way we put these electrodes on the head, and we're going to provide some effects in our epileptic animals, and I'll show how we translate this towards human-sized subjects as well. So I did say hippocampus, I believe, at some point in the introduction of this talk. And when I say hippocampus, just for those of you that are not familiar with hippocampus, in particular rodent hippocampus, we're looking at a specific subcortical structure that is connected very much linearly. If you talk about the way we use the hippocampus, you have a structure called the dentate gyrus. It has axons, in this case mossy fibers, they're called mossy fibers, axons that penetrate another region of the hippocampus called the CA3. And these neurons, these large pyramidal neurons, have axons that penetrate a region called the CA2. And logically, the CA2 has axons that penetrate the CA1. It's not important to go into the microgeometry of the hippocampus and all of the small changes in structures of the hippocampus. It's simply enough to say fibers in the hippocampus have a very strong directionality. And let's say they are from down to up. So here is the actual hippocampus of this little guy, this little mouse right here. So we would be stimulating, for example, here, and you would have afferents penetrating towards this CA1 region here, right? So these, meaning we really don't have that symmetry in this direction. And let's say in the anterior posterior direction, it's only in this mediolateral direction. And that's precisely what we used in our initial set of these orientation-dependent TI studies. Now, here on the left, these are cute pictures of mouse brains, but they're not only pictures. These are full electrodynamic simulations of mouse brain. We have all subcortical structures inside of these mouse brains. We use all of the complex permittivities of various types of tissues. Just for clarity, in this specific example, we have visualized only one side of the hippocampus, and in that respect, only visualized the CA3 and the CA1 of the hippocampus. But again, same thing as in that previous simplified cartoon slide. It's that we have neurons that are in one section of the hippocampus that send their axons downstream to other parts of the hippocampus. So here we go, CA1, CA3. In the previous experiments from Nir Grossman in 2017, that paper I mentioned, they had reference electrodes, meaning they're stimulating from, say, for example, this electrode and stimulating back towards this electrode down here. They have reference electrodes in the chest. So now that's completely fine. Traditionally, it's okay to have reference electrodes in the chest, but it does give, to some degree, a completely uncontrolled spectrum of field lines coming out of the electrode. And so it's interesting that in that initial paper, although genuinely interesting, they were able to patch neurons in the hippocampus and show that they could cause some depolarization of those neurons. They did not create any large network events. Now, in my area of research, particularly therapeutic intervention, large network events are super important. We have to have a neurostimulation method that can interface with large network events. Otherwise, we really won't have any therapeutic effect. Causing a couple of neurons to fire isn't enough to cause therapeutic effect. So what we see is we'll have a preferential orientation of these electrodes that are essentially totally parallel. We're creating field lines that are totally parallel to the axons in the hippocampus, and we'll have a totally non-preferential orientation of field lines that are totally perpendicular to the axons of the hippocampus. And in this publication, we also demonstrated that it's not necessary to use sine waves for all of this. You can also use traditional neurostimulation, square waves. I mean, in more or less all applications at this point, people are using square waves. And sure enough, pulse width modulation, the idea of mixing two high frequency or two different frequency square waves, the combination of those creates this pulse width modulated signal. And this specific envelope, this also causes the TI phenomenon. That was also a sort of a sub-result of this initial publication. So looking at both a sagittal slice and a coronal slice of this specific preferential geometry and this specific, I will call non-preferential geometry, we have this focal spot. It's very interesting. It's subcortical. We can do this and evoke it with a modulated sine wave. We can do this with a modulated square wave, no problem. So let's hope the videos work on this next slide. Fantastic. Just looking at this as an example, let's look at this anterior posterior orientation of electrodes. If we had the electrodes in this specific configuration with these specific frequencies, meaning 50 hertz envelope, this is now, of course, slowed down dramatically. We would have the electric field moving in in exactly this fashion. We would have this, let's say we're at a 20 millisecond time scale corresponding to a 50 hertz envelope. We have this electric field coming on, and we call it a hotspot. So we have the hotspot visualized as the maximum of the envelope is reached. Fantastic. And that's at that specific border structure in the hippocampus that we're interested in stimulating. The same goes for, I have no idea why this video is playing twice as fast, but it's actually better doing that. If we look at the medial-lateral orientation of this, so we see the medial-lateral, we're looking at a slice directly through along this plane. We have the same phenomenon at our 20 milliseconds corresponding to our 50 hertz envelope. So we have this beat frequency of the envelope to stimulate the hippocampus. But remember, we're on top of the cortex. We are not in cortex. We've essentially created no tissue damage. Now, I would say for intellectual understanding, this is the most exciting slide of the entire talk because nothing is happening. This is if you are using high frequency with no offset. With no offset, you have no envelope. So therefore, here is the electric field. Now, this is scrolling through time, but you have to understand this is flickering. This is flickering just like the previous slide, except it's flickering at 1,250 hertz, which I certainly can't see. So this is flickering. So then this is causing no neural activity. This is causing no firing of neurons, right, whatsoever. So we have to have that offset envelope to create that beat frequency, that envelope in the subcortical envelope. Now, in these experiments, in most of our studies, we have these very cute little mice, and we create these arrangements of topical electrodes on the mice to create these deep, this deep brain stimulation using the TI. In this specific animal, we're using these tiny micropins. And in my student's study, she avoided using any of this transcranial attenuation, although in our newer experiments we do. So she has burr holes, and these electrodes are sitting directly on top of the dura. They're sitting on top of the dura, on top of the cortex. So no penetration into the cortex, but it was just to avoid a conversation of attenuation due to skin and skull. However, as I mentioned, in new experiments, I will show results of those where it's totally topical and we don't provide the burr holes. The surgeries are very similar to any of you that are doing implanting mice, rodents, rats, whatever you have. You do the surgery, we do the implant, we give the animals a few days to rest. We're just looking for a reduction in inflammation. In our cases, it's relatively non-invasive, so inflammation is really not that big of an issue. And then we do many days of stimulation in various paradigms depending on what we're trying to study. In this specific first set of results that I'd like to show you, this is the initial set of a kindling experiment. So kindling in epilepsy is where we take electrodes and we apply a specific stimulation to begin to evoke epileptic seizures. So this is in fact the result of the first step of kindling. To create a truly epileptic animal, you would create many electrode-induced seizures. But in this case, we're just going to create one and compare the results from implantable electrodes to this topical TI method. So in this case, we're looking at a time frequency plot. We can see we have this large stimulation artifact. That's very fine. And then at approximately, let's say between 15 and 20 seconds after, about 15 seconds actually, 15 seconds after the stimulation, we have this very classic SLE, this seizure-like event. So we're recording from the hippocampus. We have a stimulation electrode, a bipolar stimulation electrode located in the hippocampus at that specific point that I mentioned on the previous slides. And there's nothing special about this. In fact, this is fantastic. It's a beautiful result. You could do this in the lab with students to teach them about seizures and kindling seizures. This type of work has been published for years and years and years and years. And sure enough, it still works. Taking a metal stick, putting it in the hippocampus, applying the correct stimulation protocol at the correct frequency, and you evoke this very beautiful seizure-like event. What we did that was novel was do the exact same thing, evoke the exact same activity, meaning using any metrics that are used in any epileptic work to try to characterize a seizure-like event. There is zero difference between this seizure-like event and the seizure-like events that we evoked without penetrating the hippocampus whatsoever. So this is essentially the very first focal stimulation event that's evoked a seizure in the hippocampus. So a deep brain stimulated focal seizure to the hippocampus without a penetrating electrode. So this was very, very exciting. Now, very exciting also because it had to preferentially use the mediolateral orientation, where we saw that we were totally parallel to those fibers inside of the hippocampus. When you perform this experiment using the same stimulation parameters with the anterior-posterior orientation, I think this is very exciting, you get zero. It just doesn't work. If the students, my students are great, they continue to turn up the stimulation and continue to change the protocol, trying to evoke something using this non-preferential orientation. Let's say we go all the way up to a 1.5 milliamps, which is very high in the case of these little mice. We still have absolutely no seizure-like event, but you'll see motor convulsions, single-sided motor convulsions within the stimulation event itself, which tells you, I mean, personally, I absolutely do not have an ethics permission to evoke motor convulsions during stimulation. So this is of course not an experiment that we can continue doing. So we obviously don't. But there are some reasons for this, and I think they're very exciting, and I'll come back to exactly C and D. But for the moment, let's investigate how A and B compare in a little more detail. So we go to the implantable experiments compared to the TI. So we can do this exactly with the implantable electrodes. We can account for orientation of electric fields. So we can take the stimulator, the implantable stimulator here, just to visualize the orientation. You could visualize like a traditional twisted pair electrode. So you put the twisted pair into the hippocampus at that same border structure that we were talking about earlier at the CA1, CA3 there. And we stay totally parallel. So again, we're going to have these parallel field lines. We're all parallel along the axons. And then we can rotate the stimulator in a different cohort of animals so that we're totally perpendicular. And we try to evoke these seizure-like events. And sure enough, this is also not studied particularly well at all in any of epilepsy research, is the orientation of electrodes during kindling protocols. It's kind of interesting because the kindling protocols themselves in literature, it's a bit like the Wild West. People have their own opinions. It's mostly based on historical precedent. And I think personally, it's because of the, let's say, the lack of control for some of these dramatically important parameters that no one previously considered to be that important. Say, for example, orientation of the stimulation electrodes. So what we see for the preferential orientation is we have a threshold for kindling these events. That's fine, but we require literally double the injected current if we have the non-preferential orientation in separate sets of cohorts of mice. So double. So it does work, but it's invasive, and we require double the injected charge. So to me, absolutely no shock that you have a non-preferential version of TI that absolutely can't do this. We can't provide this type of stimulation without stimulating the cortex on the way down. But regarding the preferential orientation of TI working perfectly, if we look at the seizure-like events in detail, let's say, for example, the frequency content, we wanted to see these beta-gamma range oscillations that are indicative of really pathological epileptic activity. They're essentially not different. They're not essentially, they're statistically absolutely not different. The mediolateral TI and the mediolateral implantable electrodes. And in all cases, these seizure-like events, there's no statistical difference between them as far as amplitude and duration, and really any metric you want to use to compare. But this is all discussed in detail in the paper. So moving along, though, so I really wanted to say something about this non-preferential orientation with respect to those axons that are in the hippocampus. I think really when what we've investigated further in some of our more recent experiments, and this is precisely the case, if we look at, say, pulse width modulated TI, so in the A here in the top right, we see, okay, here's a field profile. This is great. We've applied a stimulation. That's great. It seems to be dominantly in the hippocampus, not in the cortex. That's fine. We have a much higher amplitude of envelope inside the hippocampus than we do in the cortex. But this, because of the non-preferential orientation of the stimulation, this is not enough to evoke the seizure-like event. You could imagine this could be any event in any part of the brain region. And this is like broadly how the TI will, this is broadly a limitation of this specific configuration of TI. We'll see, okay, so it's higher, but it's not enough to evoke our network event. So you turn up the stimulator, just like my students did, as I mentioned in these figures here. So the students turn up the stimulator. We do have a significant increase in envelope, but we have at this point a non-trivial envelope in the cortex. And it just seems that with this specific setup of electrodes, not seems, it just is the case, that the threshold to evoke activity in the motor cortex in our freely moving mice is lower than the threshold to evoke these seizure-like events. So when we start to increase this, trying to create the seizure-like event, sure enough, motor convulsions for these poor little mice. So what we demonstrate is that you really have to work a little bit smarter. If you take into account the structure of the subcortical structures that you're interested in stimulating, in particular the fiber tracks from those subcortical structures, you can start to use pretty fantastic utilizations or configurations of TI to stimulate that event totally non-invasively. It just takes a little bit more thought with respect to how and where you place the electrodes. So with that said, without getting into a more complex setup of electrodes, let's stay with this medial-lateral orientation of electrodes and see what else we can do with it. I highly recommend for students that are listening, this is an absolute scientific role model of mine. This is Uwe Heinemann. This is his Nature Neuroscience paper, one of many Nature papers that he has. Uwe from 2004. Unfortunately for the scientific community, Uwe passed away a few years ago, but he is one of those members of the scientific community where you read his papers. I don't think he wrote or published a paper with an inaccurate or incorrect sentence. Or I would say if it's not totally correct, the sentences in all of his publications are as close to correct as could possibly scientifically be at the time of publication. Absolute phenomenal man, phenomenal scientist. His work really, for me, it remains gold standard style work. So what did he do in this specific publication? They were stimulating CA3, CA1, recording in the exact same regions that we're looking at in the previous slides. And in this case, they're applying stimulation, high frequency stimulation. People like to use this acronym now, this HFS, which typically means stuff over 100 hertz. And as you can imagine, we were talking about envelopes and envelopes that are too high and carriers that are too high. Obviously, they don't mean 1,000 hertz or 2,000 hertz because you're really topping out at about 180 hertz. People don't stimulate the brain with anywhere really with more than 180 hertz. But in this specific example, high frequency stimulation is going to be 100 hertz. And what he noticed is in regions in the hippocampus, specifically the CA1 and the CA3, as you begin to apply more and more electrical stimulation, ripples, these really fantastic natural occurrences that occur during our memory consolidation. This is explicitly relevant for temporal lobe epilepsy, the topic that we really wanted to talk about, and the topic of my research. These ripples, their frequency increases. And now here, when Uwe says frequency, he does not mean how many ripples you're having. He means the frequency content of the individual ripples. If you take the PSD of these ripples and you look at it, the central frequency of that specific ripple is beginning to move higher. So in his case, moving from around 180 up to about 200 hertz, actually just over 200 hertz, the central frequency of the ripple. He also saw that the amplitude of those ripples is beginning to increase. You can just imagine you're adding power to the system. You're stimulating with your electrodes, the power and the central frequency. And that, to me, that makes sense. If they're getting bigger, I mean, you're talking about increase in energy, these frequencies are increasing. Why not? That seems very logical. But then also the incidence. So this is what I meant by frequency initially. So the number of ripples per minute will also increase with electrical stimulation. So how is this relevant for TI? Let's do Uwe's experiment with TI, because I mean, you have a gold standard scientist, you know his stuff is perfect. So now if you want to prototype a new method in neuroscience, a new brain-machine interface, a new type of neurostimulation, you take someone else's gold standard paper that you know is perfect with the older technology, and you replicate replicate it with the new technology. And that's precisely what we did. We can use these modulated sine waves. We can use our modulated pulse width modulation. Specifically in the next set of slides, we're using pulse width modulation. Is TI via these overlapping square waves. And what we're going to do is we're going to compare TI from our mediolateral orientation targeting a specific region of the hippocampus versus the best you can do with low frequency stimulation. Actually, I should be more specific here. This is high frequency stimulation, but when I say low frequency, I mean we're stimulating directly from the topical electrodes with the stimulation frequency of interest. So let's say 130 hertz. Here we're creating a 130 hertz envelope in the hippocampus, meaning 2000 hertz, 2130 hertz, creating the envelope at 130 hertz. And here, directly with 130 hertz, which is obviously for a non-invasive stimulation, it's the best you can do. And you can see just from simulations, there's no way that this stimulation is going to reach the hippocampus at the same amplitude, right? So you would have to just turn this up and turn this up and turn this up. And as we learned in my experiments showing non-preferential orientations of TI, you're going to barbecue everything on the way down to the hippocampus if you do this. So what are the results of this, specifically regarding the ripples? Here we look at the central frequency that I was talking about for sham. So these are the untreated ripples. These are untreated ripples. So we see that the central frequency of this, if you look at this, just for example, take the autocorrelation of these ripples, and you look at the peak difference between the central peak and the next peak, you can calculate the central frequency of this. I mean, there's many methods to do this, right? But one example, I think it's a very pictorial example, and it's nice. We see that our ripples are exactly the same frequency as UVA's natural ripples, about 180 hertz. And we see that when we TI this, applying our 138th focal but totally non-invasive stimulation, the central frequency shifts just above 200 hertz, which is exactly the result of what UVA had. More specifically, if we were to look at this in terms of power, and we look at a PSD, we would see for sham, that's fine. We see the central frequency and we see the specific amplitude of power. But at these stimulated ripples, these ripples that we have evoked, or I would say, let's say, I wouldn't even use the word evoked. I'd say the character of the naturally occurring ripples due to the TI stimulation, this enhancement because of our neurostimulation, we see that we have an increase in power and increase in central frequency. And the increase in frequency here is statistically plotted as well. You can also see my students really like these time frequency plots where you can extrapolate and show individual ripples and how you were able to calculate what is a ripple and extract the data from the raw signals. But that's this is actually the topic of a future paper we have in collaboration with NEAR that will be out very soon. So again, as I was saying, that with the frequency, we see the frequency increasing, but what we don't see is for that low frequency stimulation, when we're applying 130 hertz directly from the cortex. Like, so again, I apologize. Actually, now that thinking about this, I really should title this traditional high frequency stimulation, meaning 130 hertz applied directly at the cortex, right? So, but we see it looks just like sham. Statistically, it just looks like sham. Nothing has happened to those ripples. And the same if we look at incidence of ripples, incidence, meaning ripples per minute, sham versus low frequency, there's no statistical significance between this. Massive difference when we're looking at the evoked, these, let's say, enhanced ripples from the focal TI stimulation. These ripples do have a significant increase in ripples per minute. So, this is really a complete reconstruction of UVA's 2004 Nature Neuroscience paper, however, using completely non-invasive stimulation to create focal deep brain stimulation. So now coming to something that is deeply important to me, this is the therapeutic benefit for this. I think that that's fantastically interesting that it works with the ripples, but let's see what happens when we take these mice and we apply a therapeutic stimulation, very much like the Santé device, or very much, well, I wouldn't say like a NeuroPace. NeuroPace is a really cool device, but it's intermittent, right? The NeuroPace is trying to catch seizures and then apply a stimulation to counteract the seizure. Our stimulation is much more in line with the Medtronic Santé device. The Santé device is providing a continuous stimulation. In their case, they're targeting the thalamus. There are many, many good papers though that show deep brain stimulation applied to the hippocampus of patients with surgically intractable drug-resistant epilepsies, so medial temporal lobe epilepsies, and they have very good results, actually, with high-frequency stimulation, namely 130 hertz, applied to the hippocampus. And so when we apply this, just for fun, we look at this video. This is a totally epileptic mouse. This mouse was suffering numerous seizures per hour with our focal TI, this DBS of the hippocampus. These mice are, for all intents and purposes, if you were just behaviorally monitor the mouse, it's not an epileptic mouse. You wouldn't know, right? So that's that behaviorally. But so that's very nice. It's always cool to show a video, but let's look at the minutia of the data. And I'm going to apologize in advance. I'm going to show one of the most chaotic slides that I have in my career, probably, but it's very exciting data, and I wanted to put it all on one slide, which is probably not a good idea. But let's just start, and I'll guide you through it. Let's start in the top left. We have pathological interictal spiking in these mice. So it's epileptic mice. They have these big interictal spikes. So when we're characterizing interictal spikes in general, we usually use, or not usually, I mean classically, there's four parameters. You have the rate, meaning the number of interictal spikes per minute. You have the area, so you have this negative and then positive portion of the interictal spike. You're looking at the area of that positive portion of the spike. You're looking at the duration, so the overall, let's say from the beginning of the negative peak to the end of the positive peak, you want to look at the duration. And then you want to call the amplitude, that drop, that drop on the spike, you want to look at the amplitude of that. So if we take mice and we have baseline, so these are the mice, they haven't been treated. They're no longer, they're not epileptic yet. And in this case, we have sham. So these mice are now epileptic. These are our epileptic mice. And we're looking at the rate, the number of interictal spikes, the number of these per minute. And then we take our TI therapy. So for example, in the video on the previous slide, that mouse is being recorded while he has his therapy. And we see that we have recovered the non-epileptic state. I mean, completely recovered the non-epileptic state. It works just like, it's as effective as an implantable DBS in epilepsy of the hippocampus, the therapeutic stimulation of the hippocampus, except it's totally non-invasive. If we apply the stimulation from the cortex, it's about as bad as you would expect from an untreated mouse. If you're looking at amplitude in this case, we have the TI stimulation here in green. We see that the amplitude is dramatically reduced. And if you're looking at the epileptic mice versus the low frequency stimulation of the mouse, it's essentially no difference. If you're looking at the duration, the area, this area here of the interictal spike, again, same thing. The area, it's dramatically different, decreasing with the focal TI stimulation. If you're looking at the stimulation directly from the cortex versus the untreated epileptic mice, they're basically the same. And as you would imagine, the duration is the same. The duration of these interictal spikes is the same if you're looking at epileptic mice versus the cortex stimulation mice, and drops significantly for the TI, the focal TI mice. Now, I will say this about ripples, because ripples are super. We have pathological fast ripples. So those of you familiar with epilepsy, you know that you have bands approximately from, let's say 80, well, everyone debates it, but you could say like 80 hertz to about 200, 250 hertz. Those are ripples. Those are perfectly healthy ripples. But there's these pathological fast ripples. There are ripples that occur in a band between about 250 hertz and 500 hertz. These ripples can occur in isolated situations, but they love to occur inside of interictal spikes. They are, we would say you have a co-occurrence all the time. Here is this interictal spike. It looks just like my sketch. It looks just like these. Here we have an interictal spike, and inside this interictal spike, if we filter out the spike itself, we have this fast ripple. It's a ripple with frequency content we see right here in between 250 and 500 hertz. If you have a lot of fast ripples, you have epilepsy. We use these in the clinic to identify epileptogenic regions, and then we extract those regions. So they're surgically resected or they're ablated using some thermal ablation method. So these are very bad, and they're fantastic indicators of epileptic tissue. So what happens to these when we apply our focal TI therapy to the hippocampus? Now, one of my students told me that I'm ridiculous for plotting this data like this because it moves positive. This is just the decreased percentage, meaning the TI has significantly decreased the percentage of fast ripples that individual animals were having, whereas untreated remains untreated. So there's no real fluctuation in the number of fast ripples. They're untreated. And in the case of this low frequency stimulation, this with the cortex stimulation, yeah, in some it's essentially the same, or we have a number of points that now have more fast ripples. So I won't comment on that in detail. We have in the publication that's coming, we have some significantly more detailed data as we go into this. And so I'll leave that for the reader in future as that publication comes out, I hope by maybe early August. But let's come to something that's that really made me quite angry. In some of our initial studies, we had submitted our publications, and you saw there's the most recent paper went through Frontiers in Neuroscience. Now, I really love Frontiers. I think they're a good journal, but everybody is obsessed with impact factors and the impact of journals. And so we had sent some of our initial data to a very nice guy at Brain. I won't say the name of the editor. He's a great guy, but a colleague of ours. But he came back and said, "That's great work, thanks. But did we see the results of Bujacki and Barini regarding the attenuation of topical signals due to skin and skull?" So he was saying that transcranial stimulation essentially being very ineffective, and that we had this new transcranial method, and that mice are cute, mice are small, but there's no way that this can scale based on the Bujacki-Barini experiments. I'm sure, actually, I think most of you probably read this. They're taking topical electrodes, they're applying low frequency stimulation, meaning they're applying 10 hertz, things like this, and they see that you really don't get anything past the cortex, right? You just don't. And in fact, 75% of everything is attenuated by the soft tissue that's on top of the skull. And that's why in this specific cadaver, they have peeled the skin back and they're in direct contact with bone. So fine, so the comment from the Brain editor made me really angry, because he clearly didn't understand that we were using these kilohertz, these radio frequencies basically, to create these focal points that are subcortical. So what I did was, and this will be the topic of another publication that we have coming very soon with Nir Grossman at Imperial, is I created the Bujacki-Barini experiment. Again, I used cadavers, but instead of applying this TDCS, I applied TI. So we have the target, which is going to be the human hippocampus. We have some electrodes that are located on the skin, so EEG electrodes. And just imagine, so we're looking left, looking right. We're going to pair E1 with E2, and we're going to pair E3 with E4. That's maybe clearer to say this here. E1 with E2 will be one stimulation pair, and our next stimulation pair will be E3 with E4. That will be the other stimulation pair. And we're hoping to target the hippocampus. So how do we do this in cadavers? Because this is what we will look at. We take clinical SEEGs, so the investigative electrodes that we use to map seizures, and the electrodes that we also, interestingly, use to ablate seizure foci. We're going to put many of these electrodes into our cadaver in the medial temporal lobe and try to target specifically the hippocampus and look at the field profile inside the hippocampus. So I've blacked this out just because I think a lot of people usually have a problem with specific cadavers, but this is the anatomy lab in Marseille. These are DS5s. These are CE marked isolated current sources. We use all of these for the TI. There's a whole army of these in this room. These are 3 of my PhD students. These are fantastic students. This is Emma, Boris, and Florian. This is Roman Caron. He's a functional neurosurgeon in my team in Marseille. And this is Nico. He runs our anatomy lab. And so we are implanting at this point all of these SEEG electrodes into the cadaver. And the results, let's take the concept of the focality, or the ratio of focality, first as we look at this. If you take traditional TDCS or low frequency stimulation, let's say 5 hertz, 10 hertz, and we just stimulate not between E1, E2, as I will with TI. Let's just stimulate the way we would with those electrodes if we wanted to hit some structure that's below the surface. Let's stimulate between, for example, these 2 electrodes, which is this is real data from the cadaver, right? What we do is we stimulate and we apply 20 milliamps. But we don't just graph 20 milliamps. What we want to plot is focality. How do you plot focality? You plot a ratio of the power at the frequency. So let's say in this specific example, I stimulated at 50 hertz. So I look at the power at 50 hertz when I have 1, and I create a ratio of 1 divided by 15, meaning I have 1 hippocampus and I have 15 cortex. And so that makes sense, right? Like, I want to see focality. If this is going to be a huge number, I have an amazing focality. If, however, I have a number divided by, let me rephrase that. If I have a number divided by a massive number, I have a very poor focality, don't I? Right? It's just fractions. If you take the number and you divide by a massive number, you end up with a very small number. So that's precisely what happens. And as you would expect, it seems, it's like redundant to perform the experiment, but it doesn't matter what you do, 20 milliamps, 30 milliamps, 40 milliamps, and the DS5s can go, the DS5s can apply hundreds of volts at 50 milliamps. And this is currents. You absolutely will burn skin. You cannot apply this to a patient, right? You don't receive any focality. You can't have focality. It's like every other limitation that we've talked about on these previous slides. You just light up the cortex as you try to go deeper to these subcortical structures. It just doesn't work, and it doesn't work in the cadavers. And we knew this from the Bujaki-Barini experiments. But in the case of TI, if we're on the skin or if we go down to the bone, we can recover as much as 3 orders of magnitude in focality. Meaning, if you had 1,000 volts per meter here, you have 1 volt per meter in the cortex. This is absolutely unheard of. This is unheard of. And I mean, you can see that this terrible focality from the traditional methods doesn't even appear on the plot because it's below 10 to the 0. So you can't even see it on this log plot. So let's look at this in a little bit more specificity, though. Here is the CT of our cadavers, the CT scan of our cadaver, and you can see numerous SEEG electrodes. In fact, it's not just numerous. This is an absolutely standard medial temporal lobe implant when you're investigating seizure foci that are related to the hippocampus and the medial temporal lobe. So we have a couple of different slices as we scan through the CT to look at the electrode placement. Here you'll see those 2 electrodes I mentioned. We were stimulating between E1 and E3 in this green pair, and stimulating between E2 and E4, the blue pair. And what we do is we just from some simulations and things, I won't get into it, to create the ideal set of parameters and the orientation of these electrodes and placement to put it in the hippocampus. But let's look at just one electrode, say, for example, in detail, the data that I was talking about in the previous slide. Like, let's look at the individual electrodes and why it works. You have the 5 hertz stimulation in this specific example. We're doing 5 hertz in the human subjects that we study. That's data that I won't show. It's not specifically mine. It's in our publication, but it belongs to Neer Grossman, and so we haven't published that yet. I won't show the results of the human study. You can read that when it comes out. That comes out, I guess, relatively soon. We'll see 5 hertz. You have nothing. It's all in the cortex. It doesn't matter what you do. You have massive amplitude in the cortex, and you don't get anything down into the hippocampus. If you use this mixture of high frequencies, you see we have a field that is measured in the cortex. It's a high amplitude, right? But it has no envelope. It cannot create activity. The envelope is too small. So this is fluctuating at 2,000 hertz, right? This is 2,000 hertz, it's fluctuating. As we progress down the SEEG electrode, we begin to have more and more envelope as they come into the correct geometric placement of the focal spot. Here, this we showed functionally stimulates the hippocampus in both this cadaver experiment, this Bujaki-Barini, and also in human subjects. So regarding the focality, though, this is a simplified system. We're just looking at an even ratio. We have the same amount of current applied from each one. It has a focal spot, and we say focal spot. There are all sorts of interesting methods that we use that are not the subject of this talk to make these significantly reduced, to make the focal spot smaller. But in this specific simple example with our 2 pairs here like this, it's essentially the entire hippocampus, but none of the cortex. So we stimulate the hippocampus, very nice. We have our functional results, super. The field profiles are maximum inside the hippocampus, great. And we have something called current steering, which is where we can then shift the focus. So here you say, everything's reduced. It's not reduced. We have, and I just haven't shown the electrodes here. The focal spot has now moved from here. It's moved frontally. And experimentally, you see this as the hippocampus returning to its previous state of activity. The hippocampus will do things that it was supposed to do without being stimulated, as we move it towards the frontal lobe. This is simply changing the ratios of currents. We can cause this fluctuation of the focal spot to shift back and forth, anterior posterior in this case. So I will come to the end. I think I have completely violated the 20 minutes that I was allocated to speak. And I will simply say, here we have our standard TI. There's all kinds of work to make this more focal that we're doing. It's very cool, but at the very least, we see right now, this is the skull, this is cortex, this is cortex, this is cortex. We begin to be subcortical, and then we have this stimulation spot. And again, this is real data. This is not a simulation. This is inside the cadaver's head. So very, very cool stuff. It's deep brain stimulation, but totally non-invasively performed, and so it's in epilepsy. And I would just say, as a hint to one of our future experiments, we've started to explore using TI from the already implanted SEEG electrodes in our patients, meaning we just essentially hijack some of the electrodes from some of the SEEG electrodes that are already in the patients, and we create these focal stimulation spots that are well off in tissue regions that are typically not available for us to implant in seizure exploration, typically because of eloquent cortex, things that are involved in core functions, or because of the amount of vascularization that's in that region. So with that, I will say thank you very much for your attention. So thank you from Marseille. But as I mentioned, we are slowly transitioning to the Karolinska Institute in Stockholm. And so I would also say thank you to you all for your attention in Swedish. And with that, I am finished.